Today we are going to talk more about immune response kinetics, but really about immune memory. And we got partway through this discussion last time, and instead of doing another sort of half and half kind of thing today, I decided to just talk about kinetics and memory and expand upon the examples I usually use and kind of go into them in a little more detail. And so I'm pretty excited because I'm getting to talk in some detail about some stories that I think are really cool and things that I really enjoy talking about. So hopefully this will work really well. And we can talk a little bit more about those details going forward. I will also tell you that the software engineering students who were here on Monday thought that that was like the most amazing thing they'd ever done in their entire lives, and they're really, really, really excited, and they want to come back and see you again, and like they thought it was awesome. So good job being awesome. So we started this conversation about immune response kinetics last time, and we learned about primary and secondary responses, and the induction of both effector cells and memory cells, particularly how the memory response will happen a bit later after that primary response has contracted. And if we have other infections while that primary response is still ongoing, those infections will be blocked quite early. We'll usually refer to those as abortive infections, where we have that infections stop before it really can even get started. And we'll see some examples of that as I give you applications of this material moving on today. And we started talking about memory responses. We can also think of a memory response as being something that we call a secondary response. So recall that with the secondary response, we are seeing our second exposure to a pathogen, in this case a virus. This will lead to a faster response, as well as a response of greater magnitude. And these cells will, in fact, be more effective as well, such that the pathogen, like the virus, will really be blocked in its replication quite a bit in terms of both how long it's going to be able to replicate and how much it's going to be able to replicate. So you will never actually see those symptoms, or you will see dramatically reduced symptoms. And in this process, we see the functions of both effector cells and memory cells. So when we have a naive B cell or T cell, that T cell or B cell can become an effector T cell or an effector B cell, as we've seen thus far. Or potentially that cell can become a memory cell. And these three types of cells, naive cells, effector cells, and memory cells, have a number of differences, some of which I told you about last time, some of which I'm going to talk about a little bit more today. So I already told you a few of the differences between naive cells and effector cells. Effector cells tend to be rather short-lived. They're quite specialized cells. They also have much higher levels of expression of many of the important genes that are used by these cells. And so, for example, an effector cell is going to have 1,000 times as much gene expression of IL-2 as in a naive cell. And so this is one of the ways that that effector cell is going to be much better at actually doing its job and dealing with pathogens and responding, because it has much higher levels of things that are needed. You can see similarly we have about 100 times as much interferon gamma transcript. And so our cell is going to be much more effective at actually doing its job of being a Th1 cell, for example, because it already has way more interferon gamma transcript. And so that's going to be one of the big differences with effector cells. With memory cells, One of the most important differences that we see, and one of the biggest differences that we think about, is this difference in longevity of the cells. And so our memory cells are going to survive for a very long term. As I mentioned at the end last time, there are memory cells that can be detected 75 years after vaccination in some patients. So a memory response is going to be able to last for a very long time, unlike those short-lived effector cells. There are other types of differences that we can see between our naive, effector, and memory cells as well. So some of them are listed on this table in the left. And we've got naive, effector, and memory cells listed for a number of these different proteins. So what you can notice is that there are some proteins that just vary between naive, effector, and memory cells. And for those of us who want to do flow cytometry to detect differences between these types of cells, those markers are really important. But some of them we can think about as having some pretty important functions. So if we look, for example, at CD62L or L-selectin and CCR7, those were the trafficking molecules that were needed to go to the secondary lymphoid organ in order to start an immune response. And you can see that naive cells have a lot of those, and effector cells have a little bit of those, because these cells are going to differ in their function of where they want to go. An effector cell wants to go to a tissue to kill pathogen. A naive cell wants to go to some type of secondary lymphoid organ. We can look at the levels of BCL2, which is a protein that's involved in combating apoptosis and helping the cell survive. And you can see that memory cells have a lot of BCL2. They're going to be able to survive for a long time because they have this protein. We can see that things like interferon gamma, granzyme B, Fas ligands are high in effector cells and in some cases in memory cells as well. So again, this allows our effector and our memory cells to have a better action against pathogens than our naive cell. You can also see that memory cells have some special proteins like CD127. CD127 is a fancy term for the IL-7 receptor alpha chain. Way back when we talked about T cell development and B cell development, I told you that IL-7 was an important survival factor to keep developing B cells and T cells alive before they had a B cell receptor or T cell receptor. Basically, IL-7 is the survival cytokine. It's the cytokine that makes cells live. And memory cells have a lot of a receptor for it because they need to live for a very long time. And so one of the reasons memory cells can live for such a long time is they have a lot of the receptor for IL-7, so they can get a lot of the survival cytokine. You can also see that there are a few of these markers, like CD62L and CCR7, where there's some weird data for the memory cells. And that is because in understanding memory cells, we have actually found that there are multiple different types of memory cells. There are different subtypes of memory cells. Two of the two kind of main types are described here, central memory cells and effector memory cells. Effector memory cells are kind of closer to being effector cells. And so we have one group of memory cells, the central memory cells, that have L-selectin and CCR7, and that go back to your secondary lymphoid organs to, again, look for the pathogen's re-emergence, while the effector memory cells stay and patrol the tissues. And so we can see even further subdivision between the idea of naive, effector, and memory cells as are shown here. One other big difference between these cells is depicted on this slide. So there are some pretty big differences between the naive cells and the memory cells in terms of the chromatin in the cells. And how tightly the DNA in those cells is wrapped around the histone proteins. And so you can see this might be what the wrapping looks like in the naive cells, and you can see that there are bigger spaces between the histones and the memory cells. So here the chromatin is rather closed in our naive cell. We don't really have signaling proteins or transcription factors nearby, and so it takes a lot of work if we want to turn on that cell and if we want to turn on transcription. We've got to get these proteins nearby, we've got to open the chromatin. It's a lot of hassle. If we have a memory cell, the chromatin in these regions are already open. There are, in fact, in some cases, signaling proteins or transcription factors already present in the cell, sitting there waiting to go. And so if this cell gets a signal, it's going to be able to turn on and act very quickly and very strongly because it's not going to be so much of a hassle to get this cell going. These cells are not going to need CD28. These cells are also going to be producing quite a bit of cytokine to help themselves survive. And there may be big differences in some other transcription factors that are important in memory responses between these cells as well, as is described in the bottom. There's one thing that I sort of danced around on one of our earlier slides. And it's actually this slide and this slide kind of contradict each other, or at least don't totally address the question very well, which is, what is the relationship between memory cells and effector cells? How do we get memory cells and effector cells? There have been many models of memory cell differentiation that different scientists have come up with. And this has been something that I've spent a lot of time thinking about. This is a big part of what I did my PhD on, and so I was really, really, really into the literature of this, especially when I was finishing up my PhD. In fact, there are two papers that I can talk about that are some of the really crucial papers. They support the opposite models, and they're both amazing papers, and they were published back-to-back in the same journal. And so even if you picked up that one issue of that one journal, you'd be like, okay, so both? So this is not something that's totally well worked out, but we've got a few different ideas for what might happen with memory cells versus effector cells. And so in this figure from your textbook, figure 14-9, it shows that our naive cell can become activated and expand into a bunch of effector cells. Most of them will die as a part of the contraction phase that we've talked about before, and some subset of them will become memory cells. And so this figure from your textbook implies that memory cells are kind of the effector cells that survive. They're the effector cells that live through. There are other models that actually say that a naive cell can either grow up to become a memory cell or an effector cell. So there's this other model, a separate precursor model, where memory cells come from one kind of precursor, effector cells come from another kind of precursor. And people have argued, and so the cell gets some kind of signal. Should you grow up to be a memory cell, or should you grow up to be an effector cell? Whereas this kind of model says that everybody becomes an effector cell and just some of them live and become memory cells. There's even this other model. This paper that supported this model is like one of the coolest papers in the entire world. It's so cool. That indicates that when the naive cell first divides, the two daughter cells, one becomes the parent of all memory cells and one becomes a parent of all effector cells. And it's the coolest paper ever. And then they had a follow-up paper that's really cool, and like the whole model's amazing. And so that kind of gets to this idea of our naive cell becomes two separate precursor cells that could go on. So, in fact, a lot of people actually support a different model of memory differentiation, which is also in your textbook. And this model says that once a naive T cell gets sort of weaker signaling, that cell can become a memory cell. And with more and more and more signals, that cell can become an effector cell. And so early after infection, we're probably giving our cells very strong signal because there's a lot of pathogen around. And so those cells are getting pushed all the way towards being effector cells. They're very differentiated, they're close to death. But if a cell gets slightly less signal, that cell may become more of a central memory cell. And in fact, those central memory cells may act as stem cells to further populate other types of memory precursors and eventually the effector cell pool as well. And so there is this idea that originally when a cell is activated, it becomes this stem cell-like memory cell. And with additional activation, it loses some of that stemness, some of that ability to self-renew, and becomes more towards a terminally differentiated, short-lived effector cell. Yeah, Molly.
Do all of these models have equal merit?
All of these models have some pretty nice data behind them. And so it's very challenging. I would say that with a lot of the literature I've seen, sort of this kind of model, it's actually described by this person named Sue Keck, is, to me, makes a lot of sense. This paper is honestly one of the prettiest papers I've ever read. And so I can give you good support for both of these models. I think it's very much an open question still. We can distinguish between these different types of cells, naive, effector, or some of the many types of memory cells that have been proposed based on their expression of different cell surface proteins. So you can see, for example, some of them are our trafficking proteins, CCR7 and CD62L. There are others, like the signaling protein CD45, or other proteins that can help us distinguish between them. And so most of the work that's done in this is done by flow cytometry with a lot of parameters to distinguish the cell types. And I used to yell a lot as a graduate student. I know you're shocked. And one of the things that we would do when I was in grad school is that we would, how are they describing it here? Yeah. So we might look at a plot of CCR7 versus CD45RA. And we would say the cells that have CD45RA and CCR7 are naive. And we would say that the cells that have CD45RA and no CCR7 are effector. And we would say that cells that had low CD45RA and low CCR7 were called effector memory cells, and cells that were CD45RA low and CCR7 high were central memory cells. So this was sort of like the hot model at one point when I was in graduate school. And I used to yell all the time about this model because if you use different flow cytometry markers besides CD45RA and CCR7, you would get a different answer sometimes about like what your vaccine or what your treatment did. And I would say, guys, this is because there's more than 4 kinds. You just like having 4 kinds because there's 4 quadrants. But that isn't real. There's more than 4 kinds. And in fact, we've realized more and more now that in fact there are considerably more subsets than the perfect 4 that we wanted. And that's why the data didn't always work out. And so we should always remember that sometimes our thinking is limited by the way we're doing our data analysis or by our techniques. And like I said, I used to shout all the time about, there doesn't have to be 4, it's because there's quadrants. One other thing that we know in terms of these memory models is that, for example, if we want to make a CD8 memory cell, one thing that is incredibly important is CD4 help. And so there were two papers that came out at the same time. They're actually both depicted in the Janeway book here. One of them was done by someone named Joseph Sun when he was a graduate student. The reason why I tell you that is because we're actually going to see Joseph Sun's postdoc work on Wednesday or on Friday, because he actually discovered something that we're talking about on Friday when he was a postdoc. So this is Joe Sun when he was a grad student. Investigators took mice, either wild-type mice or class II deficient mice, and infected them with a bacterium, Listeria monocytogenes, expressing a specific antigen called ovalbumin, OVA. The MHC class II deficient mice, if you think about it, are going to also not have CD4 positive T cells. CD4 positive T cells cannot develop in those mice because of the lack of MHC class II. So they did this experiment, and then they looked 7 days after infection to see how many ovalbumin-specific CD8 T cells were generated in the mice, and they saw that there was no difference. However, if they looked 70 days later and tried to reinfect the mice with Listeria, the memory response was completely missing. The CD8 memory response was completely missing in the mice that didn't have CD4 cells. Only the mice with CD4 cells were able to make a CD8 memory response. This was also done in another experiment with a different pathogen, this time a virus called LCMV. So in this case, CD8 cells that responded to LCMV were transferred into different mice, either wild-type mice or, again, our class II deficient mice that had no CD4 cells. And as these cells were tracked over time, it was obvious that the CD8 cells were able to persist if the mouse had CD4s, and those CD8 cells actually were lost if the mice did not have CD4s. And so this said that the CD4s were doing something to help those memory cells persist in the mice. In reality, what we know is that there are many cytokines that play an important role in the development of memory cells. And so these memory cells require many different cytokines in order to live long term and differentiate into memory cells. And so part of what may be going on with these different models, Molly, also may have to do with cytokine exposure and cytokine timing in terms of how these cells are developing. And so we can see a number of cytokines that are really important, especially for memory CD8 T cells, like IL-2 and IL-15 that can help the expansion and differentiation of these cells, and then IL-15 and IL-7 that can actually help for the maintenance of these cells and allow them to live a long time. One thing that's sort of interesting is that all of the cytokines shown here, IL-2, IL-15, and IL-7, are members of the same family of cytokines. They're all members of that common gamma chain family that I told you about before, the IL-2 family. So they all have a common receptor chain, the gamma chain, as well as additional cytokines. And just to give you an example of how someone might study something like this is from my dissertation. I was studying another cytokine from this same family, IL-21. IL-21 also signals through the common gamma chain, as well as an additional partner protein, the IL-21 receptor alpha. I had mice that were either wild-type mice that were able to respond to IL-21 or IL-21 receptor deficient mice. And I infected them with adenovirus once, and then 8 weeks later, I infected them with adenovirus again. You can see that in my wild-type mice, I saw a nice primary response. In my IL-21 receptor deficient mice, the primary response peaked close to the same place as the wild type, but you can see that it contracted really fast and contracted much further. And then you can see that the IL-21 receptor deficient mice, when I infected them again 8 weeks later, made another primary response. They didn't make a secondary response at all. And so you can see that they made a response that was identical to their primary response. They did not make a secondary response whatsoever. And in fact, you can mathematically show that the rate of change of those cells increase was quite a bit different in the wild type. So it's not just that these mice had fewer cells going into the secondary response, the cells actually responded worse too. And so this is how you would study that, and this is how you can show the importance of many of these cytokines in a memory response. You could potentially do an experiment like this and then start to look at these CD8 positive T cells with all sorts of different flow cytometry markers to see if different kinds of memory cells are being made. And that can be an important way to study this problem. One reason why understanding this process is so important is because this process is the theoretical basis for vaccines. I'm not going to talk a ton about vaccines today. Vaccines get their whole own day later. I love me some vaccines. But I am going to be giving you some practical examples of this material today, and in some cases they are vaccine practical examples. One thing to note, to realize, is that with a vaccine, what we are hoping to do is give you some type of preparation of your antigen to force your body, you can even imagine to trick your body into making a primary response to the antigen. So that if you are ever actually infected, you see a secondary response when you're infected. Thus, you make a really strong, a really good response, and you never know you're infected. You never see the pathology, or you see less disease than you would have if you had to make a primary response. And so we're trying to give you a preparation that doesn't make you sick, that doesn't cause any symptoms, that is safe, so that when you get this real infection, you are actually going to make a primary response that will be able to be protective. So all of this information is awesome. And what I want to do for the rest of the time today is talk through some specific cases of ways that we can apply this information. And some of these I was planning to talk about even before I realized we were going to have a little extra time today. Some of them I'm just expanding on. And I like a lot of these examples because they address a few different questions that people often have. So there are a lot of things that people want to know about and want to know how they work or why they work or what's going on. But what I hope that you also see by the end of looking at these examples is one of the complexities of immunology. So oftentimes, at the end of an immunology class, students will look at me and they'll be like, okay, great, get it. But what I don't get is why we get sick. Because there were so many things protecting us, how the heck are we going to get sick? And the other thing that people often get a bit confused about is why the same pathogen would lead to a different response in each of you. So if I had a pathogen and I infected all of you with an identical pathogen, that would be very mean of me. But if I did, you would all make different responses to that pathogen. Now, one reason for that, it might be something we already know, which is you all have different MHC types. But some of the implications of the memory responses that I'm going to show you here would give us some other reasons why you would have different outcomes that I want you to see. And some of the cases I'm going to show you are vaccine cases, so we can think a little bit about that. And a lot of them have to do with kinetics. I almost retitled this slide for this point of the lecture to make it say immune responses have consequences, because that's kind of what we're going to be seeing for the rest of the time today, are some of the consequences of some of these immune responses. And so the first story that I want to tell you is a story that I'm going to tell you a little bit about mumps. And I often talk about this story with chickenpox, but we have actually gotten some really nice data on it recently with mumps, and it's a little bit clearer to give the story with mumps, although I'll give you the chickenpox version of the story too. Because we've kind of, in the field, talked about, well, there's a thing that happens. And we just talked about it. And then earlier this year, someone actually published a paper proving this phenomenon that we always kind of hand-waved about, which has to do with mumps. So let's talk a little bit about mumps, first of all. So mumps is caused by a virus. It's called the mumps virus. Very creatively named. Mumps leads to some pretty severe symptoms. It is a virus that leads to something called an acute infection. So if you get the mumps, you get infected with this virus, the virus causes disease, and then you recover. The virus is completely cleared from your body. You have no more mumps virus. The mumps virus is just gone forever from your body. And you make a really nice immune response. So it's kind of like the textbook example of all of these things. So, how many people in this room have ever had the mumps? So, nobody's raising their hands. Why not? Why have you not had the mumps? Yeah, Molly.
We all, upon entrance to public school, are required to get a mumps vaccine, so theoretically all of us should.
So what were you going to say, Akuya? Yeah, so you get a vaccine, the MMR, to protect you against this. And so in theory, and in fact, you usually get 3 doses of the MMR. So you probably have something like this happening. So you're going to see graphs like this, a bunch that I'm going to draw. I can draw graphs. And the x-axis is always going to be kind of vague as magnitude of immune response. Because I spent so much time thinking about CD8 T cells, in my head it's probably CD8 T cells, but it could be antibodies, it could kind of be whatever adaptive immune parameter you want. And the x-axis is going to be time. And so what probably happened is that at the beginning, you had one mumps-specific T cell. So I drew my dot just above the axis. And it was hanging out. And then at some point, you got your first dose of MMR. And what occurred in terms of your immune response when you got that first dose of MMR? What might we call this response? We're going to get a primary response. So there's going to be a little bit of a lag. Then we're going to see an increase, and we're going to see a decrease. You can see that the amount of immune response is greater after my primary response than it was at the beginning. And so we're going to have this amount made. And then eventually, you're going to get your second dose of MMR. What's going to happen when you get your second dose of MMR? What? A stronger response. And what might we call that response? A secondary response. So now we're going to get this response that's going to be faster. It's going to be higher. You can see that, again, it's going to end at a higher place than your first one. Then, eventually, you're going to get your third dose of MMR. What's going to happen when you get your third dose of MMR? What do you think, Molly? A tertiary response. So actually, it's going to go even higher. And so it basically is going to get bumped up again, and I ran out of board to make it really high. So this would be basically what would happen. So you get your third MMR when you're relatively young. What happens to you after you get your third MMR? So if we kept going with this graph, you would actually see that this memory response would decrease over time. So we would see attrition of the response. You'd lose that response over time. And there's a lot that we don't know about that loss, how fast it happens, the details of it. But we kind of know in general that this type of attrition of this loss of the response would happen. Oh, okay, fine. Well, if we look at mumps in the United States, Here you can see mumps cases in the United States. And you can see that there's been this huge decrease since the 70s of mumps cases in the United States. But you can also see more recently, there have been little upticks where mumps starts to make a comeback. And if we actually zoom in on recent years, there have been decently sized mumps outbreaks recently. In fact, when I started teaching at Drew, there was a mumps I was teaching emerging infectious disease, and there was a mumps outbreak at a bar somewhere on the Jersey Shore. Then, and we had this big long discussion of it, and all the students knew the bar, and they were like, yeah, that place is sketchy. And I was like, okay. So there are these mumps outbreaks that are starting to be seen more and more frequently. This is actually being seen with a number of our vaccine-preventable diseases more and more frequently. Pertussis is another really good example. So I want you first to think about, we're gonna be a little bit fake here, because this is not going to be exactly perfect, but we're going to pretend we're going to look at my mom's immune response to mumps. My mom did not get 3 doses of the MMR because of when they put the rules in for 3 doses of the MMR, but we'll pretend she did. Okay? So she got her first MMR, her second MMR, her third MMR. And then let's imagine that she was like a teenager and she babysat some kids in her neighborhood. What do you think might have happened to her when babysitting those kids in her neighborhood? What might they have gotten sick with? Mumps. And so she might have been exposed to the mumps, right? From time to time. And so she might have gotten, I really need more board now, mumps again and made an even higher 4th response. And she might have gotten it again from some kid who sat next to her in school. And again and again. And so you can imagine her response getting boosted up and up and up multiple times here. And you can imagine her response being really, really high. And it being very hard for her response to ever decrease, like is shown here, to a point that's too low to respond to mumps. So maybe you have to have this much response to be protected. It's screen level response. And if you have less than that, you could get the mumps. You're susceptible. So you can see that eventually the person is going to have their mumps response go away. But if you got lots and lots and lots of random exposures in your community, it's going to keep pushing your response up, and it's going to take forever, if at all, for your response to actually go below that threshold level. But when you start to see mumps less and less in the community, people are going to have their responses undergoing attrition more and more often. And we're going to suddenly learn how long does the response last. Whereas before we didn't really know, because my mom had like a crazy high response. It was up so high that there was no way we could ever see how long it lasted. But now, you and I have never met somebody with the mumps. And so since our 3rd mumps vaccine, we've all been having our immune responses slowly decreasing in response to the mumps. And so it's possible, for example, that I now have had a long enough period of time since my last mumps vaccine that I could, if I met someone with the mumps, my response might have actually undergone this attrition to the point where I could get sick again. We started to see this in the mid-'80s, and that's when they put in the 3rd mumps shot requirement. And they said, oh, we think we need a 3rd one to really boost up people's responses more so they don't fall off. And in fact, there was some discussion in 2017 of do we need to put in a 4th mumps shot for 20-year-olds to, again, push the response up again. And so one of the reasons why we're starting to see a return of some of these vaccine-preventable diseases is because our vaccines are so good. And in fact, most of us are never getting re-challenged, never getting a natural re-challenge, and never getting our response bumped up any higher. Yep.
2 small questions.
Yes.
Is infection
So you can be exposed to antigen because many of our new vaccine modalities involve giving you antigen separate from the whole pathogen. And so you need to get some exposure to the antigen in order to push your response. It doesn't have to be the whole pathogen.
And the second one is after you've undergone attrition, if you were infected then, past the point of being susceptible to the disease again, would you regenerate that memory to some extent?
You would. You'd bump up again.
All the way to the top?
Not all the way to the top. It would be almost like another primary-ish, or maybe another secondary-ish, but it wouldn't go all the way up to quaternary or whatever you had before. Yep, Cal. Yes, and it is as you would expect. So for example, every flu death in New Jersey this year, there have been flu deaths in New Jersey this year. They've only been against in unvaccinated people. So yes, there's lots of such data. I will say that I learned from Twitter that if you get your flu shot and vote this year, then you get to eat all the ice cream you want for the rest of the year. I am in the ice cream club. I hope that y'all are going to be too. Now I'm going to give you the same process for me and for my mom for a different infectious disease. And what we're going to see, this one is more realistic, and we're going to see that I have bad shape as the result of this one. So this example is going to be an example with Varicella zoster virus, which is the causative agent of chickenpox. Varicella zoster is a little bit different than mumps virus because Varicella zoster does not lead to an acute infection. When you are infected with Varicella zoster, it causes chickenpox at the beginning, but Varicella zoster can also infect neurons and live in those neurons for a long period of time and eventually potentially reactivate. So even if you have Varicella zoster, when the chickenpox goes away, your body still has this virus living latently in its neurons. And at some point, this virus can come back. And when the virus comes back, it comes back as shingles, and you get shingles. So shingles is just when your chickenpox comes back. Yeah, Molly.
So I should probably, but
It's okay.
how do these, because varicella zoster is not a retrovirus,
Right.
so it doesn't have a latent reservoir, it can't integrate into the DNA.
It's not. It doesn't have an integrating reservoir. Yeah.
How does it stay latent without getting like ubiquitin tagged or anything?
So it's actually really interesting. The DNA, the viral DNA, stays in the cell usually as a plasmid, but does not integrate, and in many cases is not transcribed, and so is not detected. And so it's a loose, basically an episomal piece of DNA in most cases. There are some cases where you can actually see histone binding and chromatin suppression of that virus as well. Yep, Mal.
Don't cells usually have like a mechanism to like kick out like useless DNA?
Yes, a lot of the details of this we would love to know more about. Yes, Jordan.
So, is there like something that would happen that would cause it
To become shingles?
Yeah.
So let's talk about this. That's kind of what we're going to talk about. Yep, Nadia. Huh? So in fact, neurons are another part of this. So Molly was asking about the latency. Very frequently when viruses are latent, they end up going into neurons because neurons have big changes in immune responses. Neurons sometimes don't express a lot of class 1 because you don't want to kill your neurons. And so neurons are actually a pretty good place for viruses to hide. When we talk about peripheral tolerance next week, we're going to talk a little bit about some of the special issues with neurons and immune responses. Okay, so my mom had a T cell that responded to chickenpox. And then one time she got the chickenpox. And so she made a primary response to chickenpox. And then some little kid in her neighborhood had the chickenpox. and just so you know, we'll say, I don't know, this is the amount of chickenpox immunity you need. So the virus is in her all the time. So some other little kid got the chickenpox. She made a response. Then some other kid who sat next to her in school got the chickenpox. She made a response. Then she was babysitting for somebody, and they got the chickenpox, and they made a response. And then she had me, and I got the chickenpox, and I made a response. And then she had my sister got the chickenpox, and she made a response, and so on. And now my mom has never met anybody with chickenpox again. And so her response is undergoing attrition. It's going down. So now let's think about me in this response. I got the chickenpox. My sister got the chickenpox. And then I have never met anyone else who got the chickenpox because we've started eradicating the chickenpox and we have the chickenpox vaccine. So unlike my mom, my response never got pushed to be particularly high against the chickenpox. And my response is undergoing attrition as I go. And so at some point, I'm going to be below the threshold needed to keep herpes silent, to keep it in check. Varicella zoster is a herpes virus. I'm going to be below, for a long time, my immune system is good enough to keep this virus latent. So the immune system is also helping push the virus into latency. But at some point, my immune response is going to decrease. So the virus is not going to be kept in check by my immune system anymore. And when that happens, I'm going to get the shingles. My age group is like the poster children for people who are getting the shingles. I'm going to get the shingles like any minute now. And so we're seeing more and more people who are showing up with shingles because we're not having those additional boosts. Once we've eradicated chickenpox for most people, with the chickenpox vaccine, this problem's going to go away. But for now, there's this block of people, my age group is like in such bad shape, who are definitely going to get the shingles. And so that's why, in fact, they give shingles vaccines to many people. The shingles vaccine is the chickenpox vaccine at 2 times dose. It's just a double dose chickenpox vaccine. Yep, Molly.
Do different viruses have different attrition rates, and what is the cause of that?
Yes, different viruses have different attrition rates. No, I do not know what the cause is. All right, Nadia.
Maybe wrong, but usually when I see the shingles vaccines, they're to people much older than you.
Yeah.
What's the reason?
So again, typically your immune response, I kind of drew this in a way where it looks like my response is, undergoing attrition really early. I didn't draw it well enough. It takes a while for that attrition to happen. But that age has been shifting down as people are seeing fewer and fewer environmental exposures. Yep, Akuya.
So what did they use to determine what vaccines would need a booster shot and when?
A lot of it is things like epidemiological data of if people are starting to get reinfected. So in the past, some of it was done as sort of getting grandfathered in. Some of it was done as some experiments. But right now, oh my gosh, we're seeing people starting to get mumps again. Maybe we need to give another mumps shot. There's also some part of it that has to do with the type of vaccine. So when we talk about vaccines in more detail, we're going to talk about a few different types of vaccines that are out there. Some of them need to be boosted more than others. Yep.
Could someone get infected by the virus and get chickenpox as the first infection from someone who had the shingles, and so wouldn't that make it kind of hard to eradicate the virus?
I'm not sure of the answer to that. Off the top of my head, I am not sure enough to be able to give you a concrete answer to that. Okay, so I went a little bit nuts in terms of the examples I put in these slides, so I'm actually going to skip ahead to the one other example that I really want to talk about to make sure we have plenty of time for it, and then we'll go from there. And really, I'm going to kind of talk about 2 other things. So one of them is the fact that there is this phenomenon to realize called cross-reactivity and cross-protection. So we've talked about our immune response and its exquisite specificity many times. But in real life, if we have related pathogens, they are probably going to have some proteins or other antigens in common, allowing us to have a cross-reactive response. So in fact, the cowpox virus and the smallpox virus are quite closely related. You can see they have some proteins on their surface that are different from one another, and some that are the same. And it turns out that if you are infected with cowpox, you will make antibodies, as is shown here, and T cells, as are not shown here, that can react against those shared epitopes. And this will be something called cross-reactivity. This exact phenomenon that I'm showing here is the basis of Jenner's original study showing how he could make the first smallpox vaccine, where he showed that using cowpox inoculation could protect against smallpox. And it's because of those cross-reactive responses. That story is also a lie. We have now realized that he probably used horsepox, not cowpox, and there's this whole really interesting story about the horsepox instead of the cowpox, which is all very cool, but not a problem for right now. And this is also true of T cell responses, not just antibody responses. So this is a story I was going to go into in more detail. I'm not going to go into right now. But just to point out at the top, these are two viruses. They're not terribly closely related. But in fact, when they are broken down into peptides, they get made into these two peptides that are really similar. And the same T cell can actually respond to both of them. And so we can have this cross-reactive T cell response because of peptides that are similar between two pathogens, just like we could have a cross-reactive antibody response that you saw on the other slide. And I'm not going to go too much into this story, except to say that what you might want to know about this is that what you can imagine, perhaps, here is that because of cross-reactivity, your past infections can influence your response to later infections. Because if you got infected with a past virus that happens to be cross-reactive with a virus you got today, you're going to make a memory response. And your friend who didn't have that past infection is not going to make a memory response. And so you're going to be protected and you're not going to get sick. Your friend is going to get sick. And so cross-reactivity also has this idea, or this thing where it leads to the idea that past infections, your immune history, your infection history, can play a big role in determining your outcome to infections. This becomes a much bigger issue with this phenomenon that is depicted here and that I want to talk through in some detail. This phenomenon is referred to by a few different names in immunology. On a couple of the slides I'm going to show you, it's referred to as immune imprinting, but I learned it by its other name, which is original antigenic sin. And so this is the problem of original antigenic sin, as I learned it. And original antigenic sin, at least in the B cell case, you could understand pretty easily using a mechanism that I told you about last time with B cells. So you can remember that when we have a naive B cell responding to an antigen, it will bind that antigen that will turn on the B cell. If our naive B cell binds to that antigen in the presence of antibodies that also bind to that antigen, those antibodies will bind the antigen. The antibodies will also bind to the Fc receptor and turn off the naive B cell, so that only memory B cells respond and our naive B cells don't. You could imagine there are kind of similar things that go on with T cells, although to be honest, with T cells, we think it's more of a timing problem. That with T cells, the virus, for example, is cleared before the naive T cells even get around to getting activated. The memory T cells are so fast, they just get rid of the virus before the naive T cells have time to even get activated. As a result, we sometimes have this problem of original antigenic sin. And so here you can see a virus. You can see our virus has 3 different epitopes on its surface. And you can imagine getting a first infection with this virus, and you might get an infection with the first virus of strain that has A, B, and C antigens. You are going to make a primary immune response to A, B, and C. You're probably going to feel kind of sick. It's not going to be great. You're going to get better. Whatever. Now let's imagine you get infected with that same pathogen, just a different strain, the following year. Now you get infected with a version of that virus that has antigens A, B, and D. You are going to make secondary responses to A and B. C is not around to make a secondary response. And you're actually not going to make any response to D. Because these responses to A and B are so strong, they're going to get rid of the virus before the T cells have time to do anything. And the naive B cells responding to D are going to get shut off by this Fc receptor thing. So you're going to make this nice A and B memory response. Let's imagine you get infected again the 3rd year, now with a virus that has A, E, and D antigens. You're going to make a tertiary response to A. You're going to make a great response to A. But you're not going to make anything to D or E, and of course B and C aren't even present. Now you get infected the 4th year with a virus that has EDF, or DEF, because I know the alphabet. You've seen D 2 times before. You've seen E once before. However, the primary responses were inhibited in both of those cases. And so, in fact, you're going to make a primary response to D, E, and F, even though you've seen some of those antigens before. And so your response in some of these cases is biased by the previous things you've been infected with. It is shifting which types of responses you are able to make. So you are responding to your response is governed by the original antigen that you respond to. This is most frequently talked about in the case of influenza virus. Influenza virus is an RNA virus. It has 3 proteins on its surface, which are HA, NA, and M2, which are listed here. They're the ones that are actually on the surface of the virus, HA, NA, and M2. Your body makes antibodies to HA and NA, and so those are the ones that are particularly important for immunologists. Immunologists basically only care about HA and NA when they think about flu. And in fact, when flu varies, it varies at HA and it varies at NA. So the big differences between different strains are in the HA parts and the NA parts. If we look at the variation in all of the other proteins, they're not nearly as much as in HA and NA. In fact, when there is a new flu virus, we name it based on which version of HA and which version of NA it has. So a flu virus might be called H3N2 because it uses number 3, H, and number 2, N, et cetera, et cetera. And so here are a bunch of different flu viruses that exist, like H1N1 or H2N2, and on and on and on. So you've heard of a few different flu viruses. You know that flu changes a lot. So here, this figure and everything I'm going to show you is related to a particular paper. This is kind of a news article talking about this paper. This paper actually got quite a bit of press earlier this year. You can find a lot of stuff about it. But we have different predominant flu strains that infect in different years. And so we have, every so often, major shifts in the genetics of flu virus to lead to big epidemics. And so in 1918, the H1N1 virus emerged, leading to the 1918 flu pandemic, where about 50 million people died. And H1N1 was the predominant type of flu in America, or in the world, until about 1957, when H2N2 became predominant. Again, we had a bit of a big epidemic that year. That was predominant until 1968, where H3N2 became predominant. H3N2 has basically been predominant since then, until the H1N1 outbreak in 2009. Now it's kind of like 50-50 between H3N2 and H1N1. You might know about some of these. H1N1 was swine flu in 2009. Just to give you an idea, I don't know how many of you remember swine flu. It was awful. It was so, so awful. I don't want to even think about how sick I was with that. However, 0.26% of people who get it die. That's like a pretty, that's the percentage. That's way high for flu, but if you think about it, that's not that bad. 0.25% of the people die. It's estimated to have infected 1 in 6 Americans by November 2009, and in fact, the pandemic was declared over by August 2010. So, but it's still circulating now. It's still in the flu vaccine now. That's why you should get your flu shot. You also might have heard of H5N1 or bird flu. H5N1 bird flu started in 2003. Yet since 2003, there have only been 860 cases in the entire world. 454 people have died. And so this one's scary because 60% of people, instead of 0.25% of people who get this one, die. But it doesn't seem to transmit very well, so very few people have had it. And this is one's H5N1. The previous one was H1N1. There is also a flu virus right now called H7N9. It's currently in its 4th wave of infection. At the moment, it was first described in 2013. Since 2013, there have been 1,567 cases, 615 deaths, which is about a 39% fatality rate. And I will tell you that most people in the infectious disease field, if they're worried about flu, everybody in the infectious disease field is worried about flu, and the one that people are really worried about is H7N9. I feel like H7N9 is bad news bears. So this paper showed recently that it seemed like flu hemagglutinin HA proteins fell into 2 groups of being antigens. They were either sort of group 1, which were all really similar to each other, and similarity here is depicted by a dark color. So you can see all these H's are similar, and then all these H's are similar. So they said there's group 1 and there's group 2. And then they just started doing some other experiments. And this is the paper that came out of it. Got lots and lots and lots of press. And so they found a bunch of weird things. So first of all, they knew that in 2009 with H1N1, there were really few cases in the elderly, way fewer than they would expect. And they were like, well, wait a minute. The elderly probably got infected as a child with H1N1. So their original antigenic sin was with H1N1. And so they were protected when this came about. And so we're like, okay, this makes sense. They seem to be making they are biased because their first response was to H1N1. They are making the best response to H1N1. But then they also realized that people who were born before 1968 were likely to have had H1N1 or H2N2 as their first virus. And those viruses are both types of group 1. Turns out those people seem to be really well protected against the H5N1 version of the virus because it's also a group 1 virus. And these people are really, really, really susceptible to H7N9. And when they kept looking, they also saw that people who were born after 1968, their first flu was likely H3N2. And these people seem to be really well protected against H7N9 and do fine against H7N9 because their first response was to a group 2 influenza virus. And these people seem to be really, really vulnerable to H5N1. And so we're realizing that the first strain of flu that you get will impact your later infections quite a bit. And so in fact, when we think about how to protect the population against pandemic flu, we might need to think about how many people there are in different birth years who are susceptible to different things. And so if we worry about, H7N9, then we've got to figure out a way to protect people who were born before 1968. And just to give you an idea, in this paper, 75% of the variation in infection and 80% of the variation in death was based on birth year and based on what was the first flu you got, which is like crazy huge numbers for a study like this is shocking data. I thought about having us read this, although it's just a very statsy paper and not a very immunology data paper. So you can see that immune responses have consequences, and this is one of the many reasons why people really vary in their response to infection.